Hello, everybody. In this final lecture on chapter 3, we're going to talk about extracellular structures. And these are some of the pretty cool parts about bacteria. So let's get right into it. So by the end of this module, you should be able to discuss the roles of flagella, fimbriae, pili, and glycocalyx structures and identify the various flagella arrangements and discuss how periplasmic flagella differ from regular flagella. All right, so when we're thinking about our prokaryotes and their extracellular structures, they can be used for multiple things. One is to move, and so to move from one location to another. Others are to use to adhere to surfaces, and then even others can be used to confer extra protection. The first thing we'll talk about is the flagella. This here is an image or a cartoon of what the flagella looks like coming out of a gram-negative bacteria. We see that part of it is going to be embedded in this inner membrane, and then we have our cell wall, and then the outer membrane, and then you have this is where all the motor proteins are in between the membrane here, and then we have the actual uh, flagella coming out of the cell. And so you've got this hook piece and then the rest of the flagellin subunits that kind of curve off. Now, about half of bacteria that have been characterized have at least one flagella. Many of them have multiple flagella. Um, and then flagella are they're like little filaments that stick off of the cell, and they're a little bit different than the kind of flagella that they have in eukaryotes, in some eukaryotes. One of the things they have are this protein called flagellin. That flagellin protein can actually be used as a way for the body to identify an invader is there. So bacterial cells don't, or human cells don't have flagellin. And so the immune system can actually pick up on that as being not like the self and therefore mount an immune response. And these work like a rotary motor. And so they've got this rod and ring structure embedded here. And then and think about how like a motor like spins. This is what these do. As this spins, as the filament, um, or as the motor on the inside spins, this filament is just going to rotate around in a circle. And so as demonstrated by the arrow there. Um, and this is just a little small bit, but gram-positive bacteria possess two rings to secure the flagella in the membrane, while gram-negative bacteria possess four rings. So we got the two on the inner membrane, the one in the peptidoglycan, and the one in the outer membrane. While there are multiple different ways that microbes can move using flagella, one of the common ways that they will move is using the run and tumble methodology. This is especially common with what are called paratrichous flagella. And so what'll happen is if there's a stimulus that the organisms are moving towards, right? So this would be some sort of chemotaxis or magnetotaxis or something like that. So there's some stimulus that the bacteria are moving towards. And the way to think of it is that the stimulus is here, but usually there's some gradient. So if it's light, if they are moving towards light, if they're phototactic, then that means that where the stimulus is, it's going to be bright. That's where it starts. And then where, as you move away from the stimulus, it's darker. If it's a chemical like glucose, then you're going to have a lot of glucose here at the candy bar that just fell into the water. And as you go away from the candy bar, the glucose is going to become less and less concentrated the further you get away. And so that's an important concept to think about when we're thinking about how the run and tumble works. And so runs are when the bacteria are moving in a direction, and the tumbles are where they do like a little twist to kind of change directions. And so the way this works is that they start moving towards the stimulus, and then they will do a um a tumble, and start moving another way because they don't necessarily know if they're moving the right way all the time, um, or if they're moving in the most efficient manner. And so what they're doing is they're constantly detecting the stimulus, and as they get closer to the stimulus, then you're going to have fewer tumbles and more runs. When you're farther away, you're going to have shorter runs and more tumbles because what they're doing is they're trying to key in on the right direction to go. And so this is a common way that bacteria sense their environment and move towards or away from a stimulus. Um, And they can be all sorts of stimuli. I mentioned multiples. So one would be chemotaxis, like they're moving towards glucose or some other chemical, or moving away from some sort of deterrent. It could be something like phototaxis, where they're moving towards or away from light, or it could be aerotaxis, where they're moving towards or away from oxygen. In this image here, we see a capillary tube inserted into water, and they're demonstrating how um, a control versus a stimulant versus a deterrent would act, right? So we would expect that the um, in the control, they're just going to run and tumble, and they're not really going anywhere in particular. They'll be dispersed fairly well throughout the solution, but they're not trying to move towards anything. They're just going to keep running and tumbling, and probably fairly often. If there's an attractant, then they're going to run and tumble, ultimately leading them towards the attractant. And as they get closer, there will be longer runs and fewer tumbles. And for our repellent, then we're going to see the opposite. They're going to do um, more. They're going to do more tumbles close and fewer or shorter runs. And then when they go further away, they're going to have longer runs and fewer tumbles. And so you can also see kind of the demonstration of where they're going to congregate within the solution and in relationship to the treatment and the top figures. Flagella can be arranged in multiple ways. We can see a bunch of them here in these transmission electron micrographs. Um, there are monotrichous, which are single flagella, and this is if you've taken a plant class, this is something similar to like a trichome, which is going to be like a little hair. Um, these are, I think the etymology of the trichus and the trichome are very similar. So monotrichous, which we see over here, has the single. These can be polar, meaning they're on the end, or they can be offset. You have lophotrichous, which is kind of on one end where there are multiples. You have amphitrichous, where they're on both ends, and you have peritrichous, where they're kind of all over. And so, and then you get this kind of weird one, which is the periplasmic flagella. This is what you find in something like Treponema pallidum, which is a spirochete that causes syphilis. And so we've got over here on the right-hand image, you can see this cross section, and in the center we have the cytoplasm, and on the outside of the cytoplasm, we have this these flagellum fibers, and then you've got your cell wall around that. So it's periplasmic, and if you think about the periplasm, when you look over here, so for example, Treponema pallidum is a gram negative, so we could look at this guy, the gram negative cell envelope, and we see that there's this, inner membrane, and then we have a periplasmic space, then we have our cell wall, excuse me, and then we have our outer membrane. And so the actual flagella in these periplasmic flagella are going to reside inside of that periplasmic space, which is opposed to your normal flagella that are outside the cell. And because of this, these axial filaments that they have in here, they're wrapped around the cell, And so they actually cause the organism to move in a corkscrew pattern. All right, moving on to fimbriae. These are short, like kind of bristles, and they come out of the cell. They look like kind of like hairs. They're oftentimes used for adhesion. They allow microbes to stick to surfaces. They are useful in establishing biofilms. They're pretty common in gram-negative bacteria. And we have pili, which are similar or maybe exactly the same thing as fimbriae. Some people make the argument that they're longer and more rigid and less numerous, but the reality is that the proteins that are used to make fimbriae are oftentimes have homologs that are used to make pili, and so they're just seem like they're variations on the same sort of structure. um, and so I don't really differentiate between the two. I would never, uh, well, I wouldn't intentionally try to make you pick the right answer between fimbriae and pili, although there are these slight differences that some people hold to. So pili and fimbriae can adhere to surfaces, as we see here. The E. coli can use its FemH adhesion proteins on the pili, which can go to the CD48 or to this mannose or to this TLR4 receptor. Oops. Pili can be used in conjugation. So we see these two here. They're kind of connecting these two cells. This is called an F pilus for fertility pilus, and it's going to transmit an F plasmid, which is like a fertility plasmid that allows for the formation of this pilus. And so whichever cell is the donor already has the plasmid, and whichever cell is the recipient is going to receive that plasmid. Um, you have type 4 secretion systems. They're a type of pili. Um, the F pilus is one of the type 4 secretion systems, and we can see that used in multiple formats. It can be used to transfer proteins or genetic material between cells, which we see here. That's kind of what we see with the F pilus. It could also be used to transfer proteins into a host or into a organism that you're infecting. It can also be used to take up extracellular DNA or spit out DNA into the extracellular environment. And then lastly, we have the glycocalyx. It's a sticky carbohydrate. It's made up of polysaccharides. It helps the bacteria stick to tissues or surfaces. And since it's this kind of thick polysaccharide layer, it helps reduce the amount of desiccation, so the water loss. And it's going to, in some case, provide some protection from antibiotics if it's thick enough. Now, the glycocalyx can come in a couple of formats, slime layers, which are mostly unorganized and loosely associated. So you get something like this, where it's just diffuse around some cells, or in a capsule, which is a more well organized, tightly associated glycocalyx, like what we see over here. All right. So with that, you should now be able to discuss the roles of flagella, fimbriae, pili, and glycocalyx structures, and identify the various flagella arrangements and discuss how periplasmic flagella differ from regular flagella. I hope you enjoyed the lecture. I'll see you soon.